Hello, my dear students. How are you? I think you all are well now. So, welcome back to another learning lessons in my Physio-Pulse channels. Today, we'll discuss about the basic concept of enzymes as well as the characteristics of the enzymes. And moreover, we'll learn about the double reciprocal plot, how it can be derived from Michaelis-Menten equation. We'll find the clear explanations, diagram, and important concept here. Before begin, if you find this video helpful, please like, share, and subscribe to support the channel. Now let's understand about the basic concept of the enzyme as well as the that is characteristic features of the enzyme kinetics. So enzymes are both the proteins and biological catalysts. That means they are called the biocatalysts. They accelerate the specific chemical reactions in biological systems. It can alter the rate of reaction in biological process. They are high molecular weight compounds made up of mainly chains of amino acids, and they are linked together by the peptide bonds. As you know, both two amino acids when comes and contact with each other, they will form the peptide bonds. The enzymes are to a large extent protein molecules, although some enzymes are made up of RNA. So they are called the ribozymes. Many enzymes require the presence of other compounds like the coenzyme or cofactor before their catalytic activity can be exerted. Now, enzyme kinetics is concerned with the influence of the enzymes on the chemical reaction rates. We already known that the holoenzyme, that is mean the whole enzyme, that is apoenzyme, when an apoenzyme comes in contact with phosphatic group, they form the holoenzyme. Now, what we will see, enzyme when bind with the substrate, they form the enzyme substrate complex. And ultimately, it will form the enzyme product complex, then enzyme and products. This is the schematic diagram represents here enzyme substrate reactions. Now, there are so many cofactors, like the cofactors, some enzymes requires for the cofactors for their activation, and cofactors are non-proteinous part compounds, they are called the prosthetic group. Organic molecules, inorganic molecules like this, these are all the cofactors, and it may be cofactors may be the permanently attached to the cofactors called the prosthetic groups, like the vitamins, sugar, lipid, inorganic such as metal ions. And temporarily attached cofactors are called the coenzyme, and it detach after a reaction with the and may participate in the reaction with other enzyme. So if you have to distinguish between coenzyme and cofactor, then it is clearly shown here. The cofactors, they are called the permanently attached, and whereas the coenzyme, they are temporarily attached. Now to see the important prosthetic groups, and they are like the zinc, Zn plus, enzyme, carbonic anhydrase, alcohol dehydrogenase. These are the enzymes which requires the prosthetic groups. Fe plus 2, Fe plus 3, that is hemoglobin, cytochrome, spheroxin, Cu plus 2, cytochrome oxidase, K plus, Mg plus 2, pyruvate phosphokinase, etc. Like the coenzyme NAD plus, which is vitamin B3, niacin, and flavin mononucleotide FMN or FAD, they are vitamin B12, that is riboflavin. And in this way, we can say coenzyme can bind with the during the formation of vitamin or as well as enzymes. Now, these are important characteristic features of an enzyme. Like if you see, enzymes are both intercellular and extracellular catalysts. Number one, this is the characteristics. And some of the globular proteins are, and a few RNA-based molecules, they are called the enzymes. Enzymes can enhance the speed of biochemical reaction when bind with the substrate to form the enzyme substrate complex. Enzyme also lowers the activation of energy. Enzyme produces products using the specific substrate when bind with the substrate to form the product. Enzyme required in very less amount compared to the chemical catalysis or catalyst. Moreover, enzyme also can be recycled or reused, and enzyme also functions can be enzyme function can be inhibited or by inhibitor. Like some inhibitors are present, they can inhibit the enzyme functions. And enzymes are larger than the substrate molecules. Enzyme active site contains 3 to 12 amino acids. Sensitive to the temperature, pH, substrate concentration, all can be affected with the enzyme actions. And moreover, active site contains less hydrophobic amino acids. An enzyme forms the enzyme substrate complex. These are all the characteristic features of the enzyme kinetics. And if we have to see the Lineweaver-Burk double plot reciprocal equations, what we will get? Actually, in the earlier class, we understood about the Michaelis-Menten equation, that is the V0 is equal to Vmax into substrate S divided by Km plus substrate S. So this Michaelis-Menten equation we already derived in the earlier classes, and where we get the actually hyperbolic curve when V0 initial velocity against the curve of substrate S concentrations. So in this way, you can design the curve like the hyperbolic curve of the Michaelis-Menten equation. Now today we want to see, but we learned actually transformation of equation into the double reciprocal plot. Actually, this equation can be algebraically transformed into the equation that is double reciprocal plot, which is more and more useful in the plotting of experimental data. Because of that, if we have to invert these equations, then we'll get the 1 by V0 is equal to Km by Vmax into substrate S plus 1 by Vmax. So if we see this equation is, if you have to compare with the straight line equations, that is y is equal to ax plus b, then we will get the y-axis value when x is equal to 0 and x-axis value when y is equal to 0. And moreover, we also get the value of a, that is the slope, the value of slope that we will get from this equation. So like the Michaelis-Menten equation, if you have to graphically plot with the Lineweaver-Burk equation, what we will get? This is the curve 1 by V0 against 1 by S, where Michaelis-Menten equation we already learned that V0 against S, substrate S concentration. So here you will see the value of slope Km by Vmax, and moreover you will get the y is equal to 0, that is the x-axis value, and x is equal to 0, that will be the y-axis value. So Lineweaver-Burk plot has a great advantage of the allowing more accurate determination of Vmax and Km, and double reciprocal plot is very useful to determine the mechanism of enzymatic reaction. And this line has a slope of Km by Vmax and intercept of 1 by Vmax on the plot of 1 by V0, y-axis value, and an intercept of minus 1 by Km on 1 by S, that is the x-axis value. So in this way, you will get the different value of y-axis, x-axis, as well as the M. So if you have to compare equation with the Lineweaver-Burk plot, then first of all you will get the this type of curve. You will get hyperbolic curve in equation, whereas you will get the double reciprocal plot. This is this type of curve, that is the Lineweaver-Burk plot. And secondly, if you have to compare equation with the Lineweaver-Burk equation, where you will y-axis value as well as x-axis value are differ, and moreover the value of m that also be differ in between both to the equations. So students, we have already covered how double reciprocal plot can be transformed, can be derived from the Michaelis-Menten equations, and already we have also discussed what are the important characteristics features of the enzyme kinetics, and what are coenzyme, what are the difference between coenzyme and cofactors, mainly what type of inorganic substances or organic substances they are called the coenzyme and cofactors, and how specifically the basic concept of the enzymes that we already discussed in these lectures. So students, if you found these lectures helpful, do not forget to like, share and subscribe to the channel, and press the notification bell so you never miss our upcoming biology, physiology and biochemistry related lectures. And thank you for watching, and we'll see you in our next class. Thank you.